Hello, everyone. Welcome. This is the first CAE CET seminar of this semester, and it's my great pleasure to introduce Dr. Ashutosh Agarwal, who will be speaking on human organoids on a chip. So Ashu is Associate Professor of BME and Associate Director of the John T. McDonald Foundation Biomedical Nanotechnology Institute at UM. Like me, he has an undergraduate degree from IIT, and his PhD is from UF, and both of these are in material science and engineering. He's done postdocs in fantastic places, Columbia, Harvard, etc. Well, not etc., just those two. But the mission of his physiomimetic microsystems lab at UM is to develop human-relevant organ mimic platforms for discovery of therapies and drugs, and for modeling disease states, mechanistic studies, etc. The lab is supported by multiple NIH consortium grants, early stage commercialization grants from Wallace H. Coulter Foundation, and sponsored research project from industry. So without much further ado, Ashu, it's all yours.
Thanks, Pranay. So I should also say that it's been amazing knowing and working with Pranay and the new energy in the college, from the cohort of young guns has been amazing. It's what's been driving some of my interest in the work that takes place in COE outside BME. I've obviously been very well integrated into the BME family, but now I feel like COE is really kicking ass. So it's been such a welcome change for me. I've been here 8 years now, and I started in 2014, where I came fresh from a postdoc in a lab which was really just getting into the field of what's called organs on chips. So that's what you'll hear about today. And my postdoc lab essentially was one of the few that started this work in 2010 through a big contract from DARPA to the tune of $35 million. So one laboratory getting $35 million for 5 years, so $7 million a year, to go and start a completely new field. And that's sort of what DARPA, which is a research arm for Department of Defense, does. It puts in and pumps in a lot of money into a new revolutionary field, and then it kind of exits. It leaves after 5 years. It doesn't sustain those projects and leaves it up to, if there is grounds and if there's some justification, leaves it to the other funding agencies, typically NIH and NSF, to sustain the momentum. So that's what's going on. So the field really started 2010, exploded 2014, right when I was starting my lab here. And I got lucky when I got my lab here to have known all the program officers, both at DoD and NIH, who were champions of this. So I'll tell you that story a little bit, and how then the lab has been sustained through funding initiatives from NIH, essentially, and really been supported due to our excellent medical school and an amazing supply of graduate students here at COE. So I have affiliate memberships in a bunch of different departments and institutes at the medical school. But the idea and the focus of the lab essentially is driven by the problem. So this is my problem statement slide, right? So if we focus on the graph on the top left, on the y-axis are the number of new drugs that, if you're a pharmaceutical company and let's say you're a CEO of a company and you have $1 billion to burn, how much would that $1 billion go? How far would those $1 billion go in introducing new drugs to market? So if I'm a CEO in 1950, I could bring about, let's say 50 to 70 drugs or so. And if I am in 2000, I would bring only one drug candidate to market. And today it would take well over $2 billion to bring a new drug, completely new drug to market. And that's sort of depressing, right? So the y-axis, as you would have noticed, is on log scale. So this is a properly exponentially decreasing efficiency, productivity in drug R&D by pharmaceutical industry. And the numbers are ridiculous, right? So you have $2 billion worth of investment that brings a new drug, and you have 15 years of development. A pipeline that lasts 15 years. So any dent, any small increase in efficiency we can have in these systems would go a long way, both from the economics point of view, but also from a human healthcare point of view. And that's what's been driving the entire field of organ chips in general, and my lab in particular. The bulk of these costs and time happen when drugs are tested in human beings for the first time. And that stage, as we all know, are called human clinical trials. So drug companies really hate when they fail in human clinical trials. They would much rather fail before drugs hit humans. So that's called failing early or failing cheaply. And we are designing systems in my lab that can help drug companies fail cheaper and fail faster. And how do we do that? We do that by combining, and I know I'm speaking to a very non-biological audience today, which is my comfort zone. I have 0 degrees in biomedical sciences or biology. But this also speaks to the interdisciplinary nature of organs and chips. We do that by combining human-sourced cells and tissues, typically through stem cell technology, and using those human-sourced cells and tissues, both from healthy people and diseased patients, to coax them into environments so that they feel that they're still inside a human body. So we reconstruct the home that they live in so that they don't feel homesick. And if they don't feel homesick, they will give you the same responses when they're challenged with different drug entities, the way a whole human would. And that's really the hope. Now, if you're successful in doing that, let's say for one organ, we could probably extend that philosophy to other organs. And let's say now we have a mini heart and a mini lung and a mini, bones and mini testes and mini uterus growing in a lab. We can hopefully start pulling them out of the incubator where they're living, hooking them up, depending on the context of the question you're asking, and get whole body responses. Now let's take that thought experiment one step further. Let's say we do that not just from a whole body response, not just from a healthy person, but from a diseased person. So now we can start replicating that person's disease on a chip, and we can start asking what might be the cures that we can test for curing that disease, right? And if we do that well, let's maybe then take that one step further and say, let me now replicate a Black person on a chip and a Hispanic person on a chip and a Southeast Asian person on a chip and a white person on a chip. And maybe we can start discovering racial differences in the way we respond to drugs, which, by the way, is a well-known clinical phenomenon. Or now we start asking, even though Prannoy and I are both men and we are both, Southeast Asian descent, but maybe Prannoy might respond to drugs differently than I would. And can we truly personalize drug testing? So if Prannoy walks into a doctor's office, maybe Prannoy's doctor can be able to test drugs specific to the way Prannoy might respond to that drug and prescribe him a different drug or a different dose than he would describe me, right? So that's sort of the future that I'm talking about. And I'm convinced that that's where we're going. And within our lifetimes, that would be the way we do these things. So the story really started in 2014 when I joined. As I said, NIH had just come out with this challenge of helping type 1 diabetics. The only clinical cure to help type 1. So first, let me back out and say diabetes are of two kinds: the one that fat people have and the one that is genetic. The autoimmune component of diabetes is called type 1, and it develops in kids, and it often results in destruction of the cells that produce insulin. The fraction of the cells that produce insulin get destroyed by our own immune system. And the only way to cure these patients is to replace those beta cells through transplantation. So one usually has to wait for a human pancreas to be available through an organ donor. And those human pancreas are then digested, and then specialized cluster of cells called islets are taken out. And these islets are transplanted back in the bodies of type 1 diabetics. So here I'm showing you a picture from a press event where the director of the Diabetes Research Institute, after whom actually this process of retrieval of islet cells from pancreas is named, Camillo Ricordi. He's the director of the Diabetes Institute here in Miami. He is proudly showing off a picture of islets that he got from human pancreas and transplanted them into a body of a type 1 diabetic, a severely type 1 diabetic. And she was able to be free of any exogenous insulin injections. So these diabetics have to keep calculating how much insulin they would need after every meal and then inject themselves with insulin. I'm sure someone in the audience is a type 1 or knows a type 1. And so she, after 1 year of transplantation of these islets, had been completely free of doing any insulin injections, which is a great advance. And this clinical islet transplantation is really, I mean, Miami is really one of the big sites for pushing the clinical islet transplantation program forward. So now, 2014, NIH comes out with a challenge that even though this process sort of works in principle and one-off, can we improve the outcomes of this process? Yeah, if you have any questions, actually, I won't monitor the chat, just unmute yourself and speak up. I'm sorry, so the clinical program of islet transplantation is hampered by multiple things. One of them is obviously the supply of islets. Not a lot of organs become available for donation and then retrieval of islets. The second big problem actually is the islets that come out of a human body die very quickly. The shelf life of these islets is severely limited by the fact that they've just left the human body, and then, there's all sorts of enzymes within these islets that are digesting the islets themselves. They're feeling hypoxic, which means, they need that oxygen that the body was supplying through blood supply. And so these islets really need to be sustained over long periods of time. And the typical shelf life of these islets is less than half a day. So one has to line up an islet recipient pretty soon after the organ becomes available. That leads to a lot of these wasted islets. A lot of these islets that even go up for a transplantation actually don't go up for donation, do not actually make their way into a patient. So even if we just increase the shelf life of these islets, it would make a huge impact. And then if we are able to do both those things, improve the supply of islets and maintain their function, one could get then, as I keep saying, take that thought further and start asking, can we already predict what kind of immunological response this type 1 diabetic would mount against a foreign body, which are someone else's islets? As we all know, our bodies very swiftly reject anything we put in them if they don't belong to us. And this patient is already autoimmune. Her immune system has already destroyed, her own cells. And now you're doing this double insult by transplanting someone else's stuff into her. So, those three things are sort of what we're addressing in a continuously funded consortium grant from 2014 from NIH. So I'll take you through some of the engineering design. I keep saying this, the only new biological term I would introduce in this part of the talk is islets. All you need to know about islets is that they're a cluster of cells, and you can't typically break apart that cluster of cells because they all want to be together. The problem is the size. These cluster of cells are they can range from anywhere from 100 microns all the way to 600, 650, or even 700 microns. The typical size is 150 to 200 microns. Now, that size is far too big to implement the classic microfluidics. So I'm sure people in the audience know what microfluidics essentially is: the control of flow through conduits, inlet and outlet conduits that bring microliter level volumes in micrometer level of clearances. And what happens in micrometer level of clearances is that the flow of liquid is quite laminar. There's no turbulence. And that's really helpful in maintaining a well-predicted flow profile. Now, if the entities you're working with are hundreds of microns big, you have to raise the ceiling on the housing in which you make those cells live. So if this clearance is hundreds of microns, I cannot ensure laminar flow just from the geometry of the device, and that's a problem. If I have to do away with turbulent flow within this chamber, within this housing, then I have to bring in some sort of geometrical features to still ensure a non-turbulent flow. And that's the entire secret sauce behind the entire ecosystem of chips we design in the lab. Our organ chips are meant for large volume. Large volume means the clearance is about 1 millimeter or larger, and still ensure some sort of a laminar flow within these large volume organoids. And that's sort of also how I've differentiated my lab from my postdoc lab. And we've been quite successful there's a huge need for large volume microfluidics, and we've been quite successful in forming collaborations that have resulted in funding and publications just on addressing that side of the market. So having said all that, the chips typically have very simple one containment. And within that containment, there might be chamfer cuts at the entrance, which kind of force the fluid down. There might be obstacles in the flow to further ensure that the flow is directed down towards where the biology is. There might be other tricks like the exit, the outlet might be narrower than the entrance, so that kind of speeds up the flow at the exit. So we ensure that there are no fluidic dead zones right at the exit. And again, this is just one iteration. We go through a lot of different iterations through computational fluid dynamics softwares, typically COMSOL. And within the COMSOL, we solve and predict for the effect of different geometrical perturbations in the fluid inlet and the fluid outlet. We couple the pure computational inlet-outlet convective flows to biological phenomena. And the biological phenomena here is a typical Michaelis-Menten relationship of an islet producing insulin in response to glucose and its dependence, the insulin production's dependence on local oxygen. And thankfully for us, again, Diabetes Research Institute has a huge wealth of researchers who kind of worked on these problems, and we could just inherit the relationships that they discovered on the production. So it's a simple, chemical engineering flux problem. It's a classic production. It's a flux. It's an inlet flux. It's an in situ production, and it's an outlet flux. And that's all we're solving for here. And this design philosophy for large volume microfluidics was published on the cover of Lab on a Chip. This was the first paper out the gate from our lab, which was quite a nice moment for us. All right, so that was way back in 2017. And that really established and confirmed for me that this philosophy of going from a 3D CAD design, first like doing 2D simulations and predictions from geometry, taking those 2D predictions, converting them to 3D CAD using SolidWorks, and then doing in-house fabrication has been very critical. So if we can just do a design, a build, and a test very quickly within almost a day or 2 days in the lab, we can then go back and reiterate through the design, build, and test for an improved design, and then an improved design. And that sort of served me very well. So that philosophy we first applied to islets. As I said, this was the first funded grant, and maybe I'll just walk very quickly through these graphs. So this is how a test looks like when you secure islets, either from rat and mice or from humans. And all my work on this grant has been with my co-PI, Sherry Stabler, who used to be in Miami, who's now at University of Florida in Gainesville. And through both in her lab and my lab, what we've done is challenge these islets in my devices through two regimes. Regime one is you expose them to very low glucose, let's say 3 millimolar glucose, and then you quickly switch to very high glucose, and then you go back to low glucose, and you record on the y-axis how much insulin is coming out. And what you want to do is when your islets are fasting, you want very low insulin, and when you give them breakfast, you want the insulin to shoot up. But then you don't want the insulin to go back to zero, and you kind of want the islets to hold the insulin at a steady plateau, which is lower than the initial peak. And then after that steady plateau, you switch out the high glucose to low glucose. You want those to go back to zero. And that's sort of what I'm showing here. But what's very interesting here is with human islets, it clearly shows the problem that NIH wanted us to solve. The same human islets on day one do so much better in insulin production than on day 3, than on day 8, right? And so how do we solve this? So this was the next paper that just got published in, It's been a year now, but it took forever for us to get it past the reviewers. It got published in Science Advances, where instead of a full plastic chip built out of acrylic, we actually had a through cut at the floor. And then we stuck oxygen permeable material called perfluoroalkoxy membrane at the floor of the device. And we knew the oxygen transport properties of PFA. We simulated those oxygen transport properties. We stuck oxygen sensing liquid light fiber optic cables at the roof of the device. And we confirmed through in situ oxygen sensing how much of the oxygen permeation from below would supplement the local oxygen that these islets really need when they're producing insulin. And once we had our physical parameters confirmed, we then went on and did the entire testing the way I just showed you. Excuse me, go back. We would place the islets manually. We would close this clamp. We would infuse, low glucose, high glucose. We would collect from the outlet insulin into different container wells where we could test for the presence of insulin, or we could place the chip on a microscope and watch what's happening. And you see, if we have a solid plastic chip after 7 days, the islets lose their function, not surprisingly. But when we have an oxygen permeable floor, we see that the islets are functional. And you kind of see that in there's a lot of dyes we use in biology where you can stain for dead stuff and living stuff. And you can see all the red stuff is dead and all the green stuff is living. And so you can see that in the solid plastic chip, most of the things were dead. And in the oxygen permeable bottom, most of the stuff was living. And so this is the money slide. We sourced islets from a human donor. And then we could see that even after a couple of days in an oxygen permeable chip, we could get restoration of function. So how are we taking this further? So in the next sequence of grants, we are now assessing through, again, my collaborators, what kind of biomaterials can we encapsulate these islets in so that when we put these islets in the body of type 1 diabetics, these islets get shielded by that person's immune attack. That's number one. Number two, we are working with several labs that are deriving pancreatic islets from stem cell sources. So you don't have to wait for a human to die and a human to donate their organ and for the pancreas to become available for transplantation. There are many technologies now on the horizon that can make islets in the lab in a dish. And we are testing how good those stem cell derived islets are. And then lastly, so Shruti asks what kind of stain you use to classify live and dead cells. I mean, there's a whole menu of stains. It's not really, it's quite standardized. I think over here, we used what's called a calcine and ethyl dibromide kind of a stain where this dye will go into cells only when they're dead. So if their plasma membrane has been compromised, then this dye makes its way into the cytoplasm and then fluoresces red, and then the green dye goes everywhere. So that's sort of the combination here. Does that answer your question, Shruti? All right. Again, feel free to unmute yourself. Now let me show you a very compelling movie where the third application of the chip, which is quite exciting, is to predict each patient's immunological response. So here, let me set up the movie. Through collaborations, again, we do very little stem cell biology or immunology in my lab. We build devices to enable basic biology experiments. In this movie, in red, you're seeing a cluster of stem cell derived beta cells, stem cell derived islet cells that produce insulin. And the cluster of cells is living in my chip. And from the same patient from whom they derived these stem cell beta cells, that patient's islet cells, they're flowing in immune cells sourced from the blood of that patient. This patient is type 1 diabetic. So we know in theory that this patient's immune cells will be pissed off and will want to destroy the beta cells. And then we place them in my chip and we see what happens, right? And so right where my mouse is, you will see the immune cells come in towards the islets cluster and infiltrate the islet cluster. Eat and destroy the beta cells or the islet cells, the insulin producing cells, right? So now this is looping and I'll let you watch it one more time. So immune cells have made their way in. And that's a pretty dramatic destruction of that person's beta cells. But that's what's happening in the pancreas of that patient. And what I like to see, what I like to say is that these chips are sort of like a living slice of that patient's tissue, except you didn't have to cut into that patient. Yeah, does that make sense? All right, so so just to wrap up the story, as you can probably guess, when we show these data to at conferences or to collaborators or to consortium partners, they're all very excited about using the chips in their labs. And our philosophy is we have to make the chip simple enough that even a biologist can use it. So it's not meant as a pejorative thing, but we use it as sort of a call to action, that the chips cannot be too complicated. They have to be very simple. And you have to build in all the mechanisms to hide the complexity in the usage of the chip. So this design build test philosophy now has 2 more arms. The design build test that we do in the lab then has to be spun out of the lab. And we do that through spinning out companies. So the name of the company we spun out is BioVitro, that enters into B2B collaborations with suppliers of hardware. So all the chips that I showed, I didn't talk about this, are 2 pieces of Lego, like 2 pieces of plastic Lego that have to be clamped together. And they get clamped together using a mechanical clamp that's available from a company in the Netherlands. And once they get clamped, the 2 pieces also contain custom-made gaskets. So the green gaskets that you see here are these rubber elements that get compressed by the clamping action of the clamp. So once that happens, you have a fluidic seal, but the chip still has to be perfused, right, through different pumps and sources of different liquid, and the outflow has to be collected. And for that, we have a sort of a collaboration with a company in France called Fluigent that supplies the fluidic hardware for perfusing the chips. So the chips, along with the clamp and the fluidic hardware, are now living not just in my lab at Miami and my collaborators' lab at Gainesville, who are co-funded on this grant, but in collaborators' lab at Vandy School of Medicine, at WashU Nephrology, at Hebrew University of Jerusalem, at University of Pittsburgh's Medical Center, and at Indiana School of Medicine, University of Indiana School of Medicine, which is, this is pretty satisfying to see these people be able to push their science using our technologies. And I'll just give you one quick example. I won't go into details. This was a fairly biologically involved paper, but this was the lab at Vanderbilt School of Medicine. At their Diabetes Institute, Al Powers and Marcela Beresova, who used our chips to do a bunch of basic science experiments on the cells that constitute the islets in a beautiful study, which was published in a very high impact journal, where we got to be co-corresponding authors, just because they were able to use our chips to do the experiments that would not have been possible either by using animals or using simple cell culture dishes. So that's my story number 1. I could probably go and talk about the next completely different part of my lab's work, which is in cancer, or I find it better to stop at this point and maybe take any questions anyone has at this point, if that's appropriate.
Absolutely up to you.
Yeah.
Any questions? Because I definitely have a couple.
Go for it.
Which are probably more big picture, but
yeah.
Okay. So the first one, right? So, COE has quite a bit now in 3D printing with J&J. Also, we've made quite some purchases with imaging and advances, like we have a X-ray computed microtomography, SCM with nanoindentation and all that. Are these things that are useful for you, broadly speaking?
So metallic 3D printing, we have actually played with that for some of the components. The issue is the failure in the chips has to be 0 when we put like these really expensive either primary human source cells or stem cell derived cells. We want the chips to be as reproducibly, predictably functional as possible. And some of the issues we wanted to replace that clamp we buy from the Netherlands with a metallic mechanical clamp. And just small changes in the way that part was produced made a huge impact on the success of the experiment. And that's why we walked away from the metallic 3D. We would much rather spend the money and actually get like these perfectly reproducible clamps so that there's no failure. In terms of imaging, so what did you say about imaging? The SEM with the nano indentation?
So we have SEM with a whole bunch of nice things. It's one of the nicest SEMs on the campus. Actually, it also has nano indentation ability, and then Ali has X-ray computed microtomography.
Yeah, no, I haven't done much characterization yet. I haven't had much materials characterization need yet, but we should, I'm sure Alich is aware, but her lab does a lot, and Courtney's lab does a lot of materials characterization. Shruti, you're right. So just to be able to walk away from these like Dutch, very difficult to work companies in the Netherlands, we've explored, and I'll show you some, maybe I can show you some designs separately, where we had ideas of not using the magnet so much, but using, so all labs have vacuum, right? And so we wanted to build vacuum lines within the clamps so that the application of a vacuum essentially would compress the parts together. And so the issue with magnets sometimes is, let's say your clamps are being held by magnets, and then you have to place them either on a microscope, so you need the clearance, or you want to place them on some electrical characterization assay. And sometimes magnets might interfere with that. Magnets might also interfere with imaging. But you're right, we did, we went through a whole gamut of ways of walking away from this like mechanicals. Good question.
I'll ask one more actually.
Yes,
so now we also have quite some strengths in nanomaterials, intelligent materials, etc. with Sakrat and the Dean. So I know that some of these are feeding into the, engineering cures for cancer, which you may talk about later, but do you want to say something about if you're collaborating with these guys and what you have in mind?
With the cancer folks or with the?
No, in general, with respect to nanomaterials or intelligent materials, etc. Because I know Sakrat is very active in that domain.
Not specifically, again, sorry to be a boomer, not specifically for my research, but certainly it intersects well with, as I keep saying, Courtney's lab and Alich's lab in our department. I've sort of walked away. I mean, I'm certainly aware of those fields, right? Both my PhD and undergraduate are in material science and in targeting, but I just don't have, that's not what the focus of the lab right now is. It's a device lab. So I'd almost say like people in mechanical engineering would be way more, and I've had people actually who graduated from mechanical engineering in our college work as technicians in my lab. So that's sort of the more focused now. But let me switch gears, and on cancer, because I see we're at 11:34, I don't want to take up the whole hour. So I'm going to skip through cancer quite a lot and go directly to the idea of the cancer grant instead of the data. So the idea of, so we have another big grant from National Cancer Institute to solve the issue of breast cancer metastasis. And the way we do that, again, is through collaboration with our excellent medical school. So we've got breast oncology surgeons who enroll patients who are breast cancer patients. And we collect blood from these patients. And it's known that, 90% of deaths in breast cancer are not due to the primary tumor, but from a secondary metastasis. And the way this metastasis happens is there are cancer cells in the blood of these patients called circulating cancer cells or circulating tumor cells, abbreviated as CTCs, that their breast primary tumor put into the circulation. And these circulating tumor cells at some point, most of these circulating tumor cells will die and nothing will happen. But at some point, a few of these cancer cells, which are in the circulation, can exit the circulation and go towards a lung or a bone or a distant site. And at that site, most of these cells die, but some of these cells will switch and start getting cancerous again and will start causing metastases. So I'm showing here the focus of this grant, which is the bone marrow niche. And it's known that the breast cancer actually has a tendency to go towards the bone and cause bone metastases. So what we're doing with first, securing patient-derived cells, and second, by creating architectures in our laboratory, is to recreate this entire phenomena, which is a cancer cell deciding to exit the circulation, then deciding to travel towards a secondary site, and then deciding to cause micrometastasis in the secondary site. So the chip, so again, as I said, we source the cancer cells from primary patients from their blood. I rely on a technology that I did not develop, but I helped take it one step further through another paper we published in Lab on a Chip, where these cancer cells are trapped onto an array of precisely sized holes. So it's a microfilter slide onto which we run the patient cells, patient blood, and then the cancer cells are trapped owing to their larger size. And then we recover these cancer cells based on a technology I published from my lab, where these cancer cells can now be put inside these specialized fluidic chambers. And these fluidic chambers look kind of ridiculous in their complexity. And so this is the most complex looking chip that the lab has designed, where there is a bottom yellow channel in which these cancer cells will go in and come out and be in recirculation mode. So this sort of mimics that blood circulation. But then they can at some point start noticing this barrier, and this barrier is constructed of your endothelial cells, which line the blood vessels, and the perivascular cells, or the pericytes, that line the epithelial side. And at some point, these cells can decide to exit the circulation and come off, pop off on the top, where they will be in a liquid-liquid contact with another chamber where we would be culturing bone or bone marrow from human sources. So the idea is that the cancer cells will quit circulation, notice the bone, and then will start causing metastasis, hopefully in the bone. Now that is obviously a lofty goal, but already what we're starting to discover is the antigenic shift, or the shift in the nature of these cells when they start noticing just the circulation barrier, or the circulation barrier and then the bone environment. And the cells are telling us in the study we published last year that they go through a huge shift in their own nature, just like cancer cells do, when you put them into environments of that might cause them to travel, or that might cause them to divide. So that's pretty interesting. And the idea with this grant is we are hopefully taking this one step further and discovering the mechanisms by which these cancer cells decide to cause metastasis, and then discover agents that can stop this metastasis. But also the grants that I'm currently writing, we're hoping to, without cutting into a patient, keep getting that patient's cancer cells through, just a blood draw, and then proliferate those cancer cells so that we have a library of that patient's cancer cells to kind of first ask, is the cancer itself evolving in response to chemo? So one of the things that happens is, you give a certain chemo profile to a patient, and that patient does well for a while, and then the cancer starts coming back, which means that the cancer has grown resistant to that chemo regime. So we can be one step further and ask, is the cancer, what's the optimal chemotherapy regime that patient should be on? And so those are the kind of things we're doing with our cancer work. Now, another exciting aspect of this work has to do with our collaborator at Caltech and at Google's Google research arm, which is focused on development of deep learning modalities, where we're developing new, so this might interest you, Pranay, we are developing new kinds of imaging capabilities, which essentially are able to take a bright field image and ascribe to a bright field image false colors after having learned from a huge training set what might be a cancer cell and what might be a non-cancer cell. So this is in collaboration again with Google, and Google is donating all of its, both computational resources and manpower for free to this project, because they have a deep interest in perfecting and adding more sophistication to their own computational algorithms. And all they're in need of is like a huge, well-annotated training data set. So it's a pretty interesting conversation every time we talk with Google on deep learning, because they always come back and tell us things that might be possible. Let's just try it out. And we never even thought that you could pick up a cancer versus a non-cancer cell without staining for different markers. But that's what Google's helping us do. So that's the deep learning collaboration on cell detection with Google, which is also now a nice spin-off of this grant. So those are the two funded stories in the group. Then there's a bunch of unfunded work in the group. And perhaps the one I should, in the interest of time, focus on is the work on neuromuscular junction. So this is completely unfunded. The other work is sort of funded through fellowships that my students have gotten over the years. But this is absolutely not funded. And I say this because I keep saying this because it's often the most interesting work in the lab that finds the hardest time, that has the hardest time in finding funding success. So absolutely being run by my own sort of we're bootstrapping this project, but this is by far my favorite project in the lab, because this combines a lot of what organs and chips should be doing. So again, I get a call from a neurologist here at Miami who has just joined after having a successful practice in Brazil, who has had, whose claim to fame is working on this very rare neuromuscular junction, this disease, which none of you have heard of. It's called CMT, Charcot-Marie-Tooth disease, named after three clinicians who discovered it, CMT. And patients from all over the world actually fly to Miami now who have this disease, CMT, to be seen by Mario, Dr. Sapota. So Mario, what he does is, first of all, the disease manifests itself by loss of sort of function in your digits and then in your arms and legs and sort of like ambulatory issues. Now, what he's done is created a library. So he takes after patient consent, he takes a sample of those patients, skin cells, and he converts those cells into that patient's neurons. So he has a whole library of cells frozen down from patients who suffer from this terrible disease, CMT. It's very rare, and the mechanisms are not known, and obviously there's no cure. He can only manage those patients. So what he and I have been doing over the years is building towards designing that the same patient's muscle, putting Mario's neurons from those patients, and designing architectures where the neuron is able to extend what's called axons down towards the muscle, so that now we can electrically get the neuron to fire. When the neuron fires, we watch how that propagation takes place down the axon. So axon is sort of this electrically conducting, think of this as the wiring between the source, which is the neuron, and the target, which is the skeletal muscle. Like any muscle, skeletal muscle that we move is getting moved because some neuron is telling and stimulating that muscle to move. And the way that stimulation happens is the neuron initiates an action potential, which travels down the axon. And then it physically connects to a skeletal muscle. And right at the connection, which is called a synapse, it releases some small chemical molecules called acetylcholine. And the skeletal muscle has receptors for acetylcholine, which is a fancy way of saying that the skeletal muscle is able to detect small changes in the signaling molecule. And when that signaling molecule is present, the muscle twitches, right? So all the motion we're doing with the skeletal muscle is because the neuron is firing. That firing action potential is traveling down an axon and letting the skeletal muscle know to move itself. Now, I'm going back to the disease. We know their skeletal muscle is losing function. And we have, Mario knows it's a neuromuscular junction disorder. So what we are able to do now with this platform is ask whether it's a disease of transmission or it's a disease of connection, right? And that itself would be new knowledge in the field of CMT. And the fact that we're able to source cells from the patients who suffer from this rare disorder is the perfect application scenario in my mind for the field of organs and chips. I think organs and chips will not solve or discover drugs for like cardiovascular disease, but I think organs and chips can be directly applied for either discovering disease mechanisms or discovering therapies for these rare genetic disorders like CMT. So of course, it builds in small steps. This work we started in 2016 or 15, and it took us a long time just to do the basic stuff. And the basic stuff was first getting both neurons and skeletal muscle to be happy on the same surface, which we published in this pretty nice journal. And then getting the neurons and muscle to be happy in the same liquid medium. And that itself took a long time. And that was published in another paper 2 years ago. And then we have a bunch of data where we're able to ask, are the neurons happy along with the skeletal muscle? And we do that through these microelectrode arrays, which can record small field potential changes when neurons fire. And then we are able to get both the neurons and the skeletal muscle in green here to be, alive and happy on the same surface and also in the same liquid media. We are able to ask, so look at these like beautiful images of neuronal bundle clusters extending axons, these thick axons down where the skeletal muscle is. And if you take a zoom in into the skeletal muscle in green, we see all of these nerves right in the vicinity of skeletal muscle. We're also able to pick up nerve endings right onto a muscle and then stain in red here those acetylcholine receptors. So these are those receptors, the sensors for the chemical called acetylcholine that the neuron releases, right? So the assembly of acetylcholine receptor tells us that this muscle actually detected that there was a nerve ending and it assembled these sensors right where the nerve was ending. And we're able to also stain for the vesicles, the synaptic vesicles in white here. So, we have as good a proof as we can get that the skeletal muscle and the neuron is talking to each other. And then the actual money graph is this one, the bottom right here, where the size of that acetylcholine cluster is significantly smaller when the cells come from patients with the disease rather than when the cells come from healthy people, right? So you see a sharp reduction in the ability of the skeletal muscle to assemble the acetylcholine receptor cluster when it's from diseased patients. So that's very exciting, and that's never been done for this disease. We are quite happy to take this further. And we're writing grants together, and hopefully one of them sticks. But that also speaks to the crapshoot of what NIH funding is in terms of the most compelling stories from the lab not getting funded. All right, so maybe in the last 10 minutes, I will give a general sermon of where we're going next. I think the real power of organs on chips is revealed when either we work on these rare genetic disorders like CMT, or when we start coupling organ 1, 2, 3, 4, 5, 6, 7, 8, and get whole body responses. And I think coupling these organs together is very important. There's a lot of very potent scientific and technical challenges that throws up. One of which I alluded to before is just the same liquid that all of these organs would be happy in, right? So we all of us have blood in our bodies that's perfusing all the organs, and they all seem to be happy in that blood. But there's no substitute or replica of that blood for doing experiments on a dish. So one would think that we would have figured out a universal blood substitute for doing experiments on a dish, but we haven't. From a mechanical engineering standpoint, having all of these organs talk to each other also means that you have to figure out all the pumps and the valves and the pressure controllers and sources of leakage and sources of introduction of air bubbles, all of the sterility failures, which is not trivial at all. I think 90% of the time of my graduate students is spent in just solving the stupid non-biological failure modes. From a engineering point of view, we also don't know how to scale a lung chip and a heart chip and a kidney chip and a skin chip and a brain chip, right? We know the human body has scaled the lung, heart, muscle, skin to each other, but what are those scaling laws that can be interpolated or extrapolated to a chip format, right? Do we scale them by the weight or the area or the rate of diffusion or the amount of metabolism? We don't know, and no one knows. And so that's another discovery waiting to be happened. And the last one is, if I do get a response from my chip, how do I know what that response translates to for a full human being? So, again, no one knows this, and very smart minds are working on translating or extrapolating a response from a chip to the response on a human being. From an engineering translational point of view, we also don't know whether industry will take these devices and adopt. And I think quite deeply about these issues. One of the things is the industry is very comfortable with using, fairly standardized, very high throughput, very cheap, but very stupid systems called these multi-well plates. My organs and chips right now are super sophisticated, very costly, and the throughput is very low. So I think there needs to be, and I keep stressing this, we need to give up some of the sophistication and gain the we need to win on the cost axis, and we need to win on the throughput axis and the simplicity axis, so that these devices don't scare off pharmaceutical industry and we're able to use them. So one of the challenges we solved in that was, you cannot design a chip and then ask a pharma company or a basic biology lab to adopt this architecture of fluidic control, right? That's what it takes to run one of these chips. And no one who does basic biology is going to be comfortable with their cell culture area to look like this. So what we worked towards, and this was a senior design project made by 4 senior design students in BME, who worked for 2 years actually on this and got the best COE senior design award, was they worked on a fluid handling system, a fully integrated fluidic handling system, where they're able to perfuse the chip, switch between different sources, and like let's say switch from a yellow or an orange source to go to a recirculation mode, and then from a recirculation mode back out to a green source, and control. So they basically bought all the hardware from original hardware, and they programmed all the hardware to sit together. They constructed this housing, and they wrote the GUI for all of this hardware, and this got published in a very nice journal actually. So I will stop there, and I will say that this is the cancer diagnostic company that I'm affiliated with, and this is the organ chip company BioVitro that's serving some of the collaborators right now. And I think it was Shruti who asked, can we use magnets? Shruti, this is the design we had for the vacuum pump, right? So these 2 clamps, instead of a mechanical clamp, could be connected to a vacuum source, so that these 2 pieces clamp the chip together. And so, yeah, we're working towards different clamp designs. So I'll stop there and say we have a pretty nice, diverse group of students. All the work I've shown so far is actually work from people who graduated, who are in italics. And then we have a fairly new group of students. We've been very lucky with having amazing collaborators at the medical school, funding sources from both foundations, pharmaceutical companies, and NIH in the form of the cancer grant, in the form of the diabetes grant, a diabetes fellowship that my previous PhD student had, and a diabetes R01 that my collaborator has, who's funding one of the graduate students in the lab right now. So I will stop there and take any questions, Pranay.